Yeah, yeah. They're dribbling in. So I can see people are slowly drifting back from coffee. I think we ought to begin because we're running 6 to 7 minutes over, and I know that there are discussions planned for the lunch break as well. So we had two great talks this morning on genetics, and now we're going to turn our attention to thinking a little bit more about what's going on in the brain, and in particular thinking about the relevance of neuroimaging in understanding ADHD. We have two fantastic international speakers who have kind of really led developments in the field. First of all, Adriana, who will talk about similarities and differences between autism and thinking about overlap in the brain connectome in these two related, if you like, certainly at the genetic level, as we heard, neurodevelopmental disorders. And Philip Shaw, whose name I spelled incorrectly on this thing, is going to think about developmental outcome studies, and particularly thinking if you've got children with ADHD and they persist with ADHD or they remit, and so what are the brain kind of processes that are kind of tracking this clinical disorder through development. So just by way of introduction, I just wanted to give a brief conceptual perspective on how to think about neuroimaging findings. I'm not a neuroimager, so I don't pretend to have any sort of in-depth knowledge about which are the key circuits involved in neurodevelopmental disorders. But just to think more generally about the kind of things Steve talked about and Joel Nigg talked about, really the complexity of the relationships between genes and environment. I mean, what we're all after at one level is finding out what is going on in the brain, driven by these genetic and environmental risks leading to behavioral outcomes, to disorders, and to traits. And I think we've learned that at the genetic level and at the brain level, there's no neat one-to-one relationship. I mean, there are going to be common risk factors leading to a range of correlated comorbid traits and conditions, if you like. But for a long time, people have sort of focused on this sort of very linear endophenotype model, where essentially you're thinking of a set of genetic risks that in some way these are expressed through cellular function and brain function, patterns of neuronal activity, cognitive deficits, and then leading on to behaviors. And I think what we've learned, or what we're learning, is that at each of these different levels, it's a very complex story. And I mean, just at its simplest, we used to think that maybe there'd be single genes leading to single outcome effects. We now know that really everything we're talking about is polygenic. And so we heard that this morning. But the other big aspect which also comes out of the current age of genetic research is thinking about pleiotropy, where either single genes or these polygenic effects actually don't have one outcome. They actually have multiple unrelated outcomes, or sort of correlated outcomes, but not causally linked outcomes. And this is called pleiotropy, the multiple different outcomes of gene effects. And you can imagine that at the brain level, we're interested in identifying which of these neural processes. But in fact, what we're really just measuring at this point are correlations or associations. And we have to do a lot more work to find out which are the causally relevant processes. So at the top, it might be that one gene or set of genes affects one brain circuit or one brain region, and that brain region perhaps has just one particular behavioral outcome. But it could also be that one set of genes affects many different brain regions, and each of those brain regions or neural circuits has its own behavioral outcome. But of course, it's much more likely that sets of genes affect many different processes in the brain, and each of these processes also have their own multiple different outcomes. And so really the whole thing is very complicated. And so how do we unravel this? And I'd point many of you back to this, what I kind of think was a seminal sort of really nice paper that summarized the thinking in this area from Kendler and Neal on the endophenotype concept. And at its simplest, we're trying to separate out what we think of as causally relevant brain functions that mediate between genes and behavior from these kind of pleiotropic, what he called genetic risk liability markers, which is kind of a pleiotropic model where the genes have these multiple outcomes. And I think for some outcomes, everyone is already thinking that these are probably pleiotropic effects. So you have a common genetic liability that leads to ADHD, but it overlaps a lot with many other outcomes like specific learning difficulties or autism. And we're very happy to think of those as discrete disorders, even though they share a common genetic basis. But somehow when we come to think about cognitive functions, because they're somehow linked more directly to the brain, we immediately assume that these are causal. But of course, they could also be pleiotropic effects, what I would think of as neurodevelopmental comorbidities, if you like. And of course, the same thing applies when we measure things in the brain. So it's very hard for us to really pull out what is causally related and what I would think of as a kind of neurodevelopmental comorbidity, however we're measuring it. So I just wanted to put the talks we're going to get within that kind of framework. And so we're going to begin with Adriana, who will talk.
Just see if I can do this. Sorry. There we go, that's good. Oh, it's working. I'm just getting my little notes page. Good morning, everyone. It really is a great pleasure to be here, and I'd like to thank the organising committee for the opportunity to come and talk with you all. I have no conflicts of interest. I'm funded by intramural NIH, and I'm still open for business. So to just give you an overview of this talk today, first I'll describe remission and persistence in ADHD and consider why it's so important to study it. I'll then outline some of the conceptual models that really others have used to understand remission and persistence. Then I'll describe 4 studies we've done in my group, looking at the associations between adult remission and brain structure and function. And then I'll attempt an integration with some of the great prior literature. So first, however, I'm a clinician, I'm a child psychiatrist, and I thought I'd tell you about 2 of the kids I've seen in our study. So little Peter came to this study. He was 6 years old. He was accompanied by his extremely tired-looking mother, who said, you know, this is the most hyperactive child in the world. He came into the room. The chairs in our assessment room looked rather like this. He immediately managed to get his head stuck in one. We extracted him. He found the plug. Anyway, it all went on. So we managed to get him good and tired. He did about 2 hours worth of neuropsychometric tests, and then very, very kindly fell asleep in the scanner, which is really excellent. So thank you, Peter. He was really there, a lovely kid, really nice kid, but completely unable to play reciprocally, unable to let others finish their sentences, hopelessly inattention, really struggling at school, really struggling with his peers, and his mum was really struggling with him. He was impaired in multiple domains and was appropriately diagnosed by his pediatrician as having ADHD. Susan was likewise described by her mum as being very hyper, having a Duracell battery in her that never ran dry, extremely physically impulsive, unable to sustain attention on anything, even tasks that really quite interested her. And her first grade teacher described Susan as being a bright kid, but almost unteachable by virtue of her extreme inattention. And she was failing to learn. She too was diagnosed with ADHD. So fast forward in their lives, and how do these kids fare? Well, Peter received and responded well to standard treatment, a combination of psychostimulant medication and accommodations at school. And by middle school, he noticed his symptoms weren't really that bad. Occasionally, he'd forget to take his medication. He didn't notice that much difference, and so quite appropriately, he stopped it entirely. And by the age of 25, when we last saw him, he was doing beautifully. He had graduated college, he had a job that he absolutely loved, and he was essentially symptom-free and enjoying his life. By contrast, Susan, she actually did quite well in primary and middle school, again on a combination of medication and school accommodations. However, in high school, she hit a wall. As academic demands became greater, her inattention became exceptionally difficult for her to cope with. She began to fall into a vicious cycle. She got despairing at school, started to skip school, barely managed to graduate. And when we last saw her at the age of 24, she had a very marked constellation of symptoms which were very, very impairing for her. As a result, she said they were the main contributors to why she was failing to sustain employment and to sustain meaningful relationships in her life. Now, I hope Peter and Susan illustrate the very variable clinical outcomes of ADHD. These kids look pretty similar as kids. As adults, they have quite different endpoints. So how frequent are these different endpoints? Well, here's Steve Ferrone's very lovely meta-analysis of studies up to 2006. The green line in this graph shows the proportion of kids who continue to meet full diagnostic criteria for ADHD as they grow up. If you look at the x-axis, you can see at around age 20 to 25, it's about 15 to 20% of kids will continue to have the full syndrome. The proportion of those with impairing symptoms that can really get in the way of life is much higher, at about 65 to 70%. I actually looked at 6 recent clinical prospective follow-up studies, and these estimates are holding out very well. They're a little bit higher, I think, because the newer studies are clinic-based, but they're pretty much a consonant with the earlier findings. So it's important to study outcomes because ADHD persisting into adulthood is common, and it's a major public health challenge. And understanding the processes that underpin different clinical outcomes might help provide biomarkers to help us predict these outcomes. And finally, understanding how remission occurs in some might give us new treatment ideas so we can accelerate processes of remission in all. So how do we do this? Well, of course, the absolute gold standard for looking at neural factors in remission is to do an experimental intervention to show that altering a neural process leads to remission. This is exceptionally difficult to do, very rarely done, and as a result, the vast majority of studies are observational. Now, causal inferences aren't possible from observational studies, but they can point powerfully to candidate mechanisms. So in observational studies of this nature, the ideal is to prospectively acquire both clinical and neuroimaging data together from childhood into adulthood. And of course, this hasn't been entirely possible, as many of the newer imaging techniques used, for example, to map brain connectivity, are relatively recent innovations. So as a result, most groups have examined cohorts that have been followed clinically from childhood but had their neuroimaging only at the adult endpoint. And what can we reasonably infer from adult endpoint studies? Well, a popular model of remission is that children who are remitting develop strategies to compensate for their symptoms, and these compensatory strategies are tied to underlying neural reorganization. Now, by this model, the remitted brains in adults will not look like the brains of individuals who were never affected, nor will it look like the brains of those who have persistent ADHD. Rather, there will be unique features here, unique features of neural reorganization that are beneficial. An alternative model is that remission is linked to a correction of early anomalies of brain structure and function. That is, there's convergence with remission towards typical dimensions. Now, by this model, the remitted brain will look essentially typical, like the brain of the never affected. Now, remember, just to go back to my caveat, when imaging is acquired at the adult endpoint only, this assumes that there are anomalies in childhood that are later corrected. I think this is a reasonable assumption given what we know about the childhood ADHD brain, but it's an important caveat to bear in mind. And the final model, here I'll call it the fixed trait model, argues that neural anomalies of childhood ADHD leave an indelible mark in the brain which persists into adulthood regardless of clinical outcome. So of course in this model, remitted and persistent brains will both show similar atypical features despite their very different clinical presentation. Now these models are certainly not incompatible. For example, a lot of this work is really driven by the work of Halpern and Jeffrey Newcomb, and they have argued that the onset of ADHD is caused by subcortical anomalies that will persist throughout the lifespan. However, the variable clinical course is held to be determined by the plasticity of the cerebral cortex. So the cerebral cortex will reflect outcome, the deeper brain structures will reflect childhood history. I'll again stress all of these models make similar predictions about what the persistent brain will look at, but make quite different predictions about what the remitted brain will look like. So to test these different models, we looked at a group of children who were enrolled into ADHD imaging studies in the late 90s and 2000s at NIH under Judy Rappaport and Javier Castellanos. So at this time, the kids had a mean age of about 10. When I joined NHGRI, we brought them back to NIH. We managed to get about 60% of them at a mean age of 24. Now, we acquired anatomic MRI prospectively, because it's been around a long time, but we imaged the brain's structural and functional connectivity, only at the adult endpoint. So adult outcomes were ascertained through clinician DSM-based interviews done by me. I used the CADID for ADHD and the SCID for Axis 1 disorders. We emphasize data both categorically, contrasting persistent against remitted, and also dimensionally, regressing all the neural features against symptom counts. Most of the slides I'll show will give the categorical outcome results because they're a bit easier to understand, but they're always backed up, unless I say otherwise, by the dimensional findings. So in this talk, I'm going to say mainly about the connectivity imaging that was acquired at the adult endpoint only. However, neuroanatomic imaging, as I said, has been around a long time, so many of these kids had anatomic imaging acquired in tandem with clinical assessments from childhood into early adulthood. So we're combined these data together, and we can ask where, for example, is change in the cerebral cortex associated with different adult outcomes of childhood ADHD. Here we looked at the thickness of the cerebral cortex in 274 participants. So we found that differences in the adult outcomes of ADHD, specifically inattention, were associated with different neuroanatomic trajectories during adolescence in multiple cortical regions. They were mainly prefrontal. These regions, some of them are shown here in a medial view down the middle of the brain, and you can see here this is a lot of cingulate area, and the regions that were hit lie within things that Adriana was talking about, the default mode network, the cognitive control network, and some of the attention networks of the brain. So these graphs show what was happening here. Just to orient you, the y-axis is how thick the cortex is in those colored regions. The x-axis is age. The blue line are the never-affected comparison group as they get older, so the cortex in this region gets thinner. Look at the green group, who are the remitted ADHD. You can see they start off a little bit thinner, but as they get better, so they converge towards more typical dimensions. And by contrast, here are the persistent group as they persist, so there's a persistent deficit. Now, one thing we were very interested in, anatomy is great. One thing we're learning from it is that the effect sizes, the differences between the groups are going to be small, and we're seeing that very clearly from Enigma and big multi-site consortium. So many are looking at other modalities. The concept here is if we get lots of small to medium differences between big groups, perhaps we can start to think of clever ways of adding these together to do something useful with the data. So we looked at white matter microstructure. So we did so partly inspired by Adriana's paper, actually, which said that ADHD should in part be conceptualized as a developmental disconnectome, as the end product of disrupted connectivity within multiple neural systems. Now, I just mentioned how in the anatomic study, we found that different adult outcomes were tied to different anatomic trajectories of cortical networks. A logical next step is to examine the properties of the white matter tracts that connect the different regions of these cortical networks. And to do this, we acquired, as Adriana said, DTI on 149 subjects. Here we contrasted remitters, persisters, and the never affected. Amongst the many data processing steps, all images were visually expected. We excluded subjects with more than one bad volume, along with those showing even minimal movement. I'll note the outcome groups didn't vary in any imaging parameter, including residual motion. We still took it out along with the effects of age and sex, and all the analyses I'm going to show you are corrected for multiple comparisons. So here, we're looking at the microstructural properties of the 11 white major tracts in the brain, which you can see in the cartoon there. This is what we extracted from the DTI images on all subjects. So what did we find? Well, in several tracts, there was a difference between the adult outcome groups in the measure Adriana mentioned, fractional anisotropy, which is a measure that reflects white matter microstructure. The results for one tract, this is the inferior frontal occipital fasciculus in blue, is shown there as a box and whiskers plot. As you can see, the never affected control group and the remitted did not differ from one another, but both differed from the persistent group who had a lower fractional anisotropy. And this pattern, the remitted being similar to the never affected group, held across nearly all tracts. Only the persistent group differed. Next, we reanalyzed the data on the basis of the childhood history of ADHD, combining the persisters and remitters and comparing them to the never affected, and here no significant differences were found. So in terms of the competing models of remission, the results are more compatible with the concept that remission is due to a convergence with the correction of possible early anomalies. There was no evidence here of lingering changes to white matter microstructure that reflected earlier childhood history of ADHD. So next, we looked again at another connectivity, the brain's intrinsic functional connectivity. As Adriana has shown, we defined functional connectivity through the patterns of coordinated brain activity that occur while the subject is simply at rest, free of any explicit task demand. Now, usually, as in Adriana's work, this is done using fMRI. It's also possible to measure neuronal activity using magnetoencephalography, and I'm going to focus on this modality. You can see here, this is the MEG machine that we used. This technique non-invasively picks up magnetic fields that are evoked by populations of neurons firing. This allows a relatively direct observation of neuronal oscillations across the brain that synchronize activity that scaffolds cognition that's really pertinent to ADHD. MEG, like EEG, also has exquisite temporal resolution of the order of milliseconds, which complements the very nice spatial resolution we have in fMRI. So, to reiterate our central question, what will the remitters in this group look like? Will they be similar to the never affected as we found for white matter microstructure, or would there be evidence of compensatory reorganization of the functional connectivity? Or would remitters show the same anomalies as persisters, reflecting their shared childhood histories? So prior to the MEG analyses, all the data were inspected. We excluded epochs that showed movement artifacts or muscle artifacts, and so we excluded about a third of the data. We then filtered the clean data onto one of the 6 frequency bands in the brain, and the signal was mapped onto a cortical grid, and this cortical grid had 2,000 points across the cerebrum. For fMRI, we used the techniques that Adriana's described, 6 minutes of resting fMRI. We excluded volumes that showed more than minimal movement, 0.2 millimeter, and we regressed it all out along with age and sex. So using these data, we defined connectivity patterns that capture interactions within and between the brain's major functional networks. Now, to do this, we calculated all the pairwise connections between the 2,000 or so points across the cortical grid. Each one of these cortical points was allocated to one of the brain's major functional networks, such as the default mode network or one of the attention networks. Thus, we knew the network from which each connection began and ended. So we next extracted from this complex data connectivity patterns that were stable, and they're stable in the sense that they consistently emerge over many bootstraps of these data. And we found 56 of these stable connectivity patterns in MEG and 6 in fMRI. And here's one of the fMRI stable connectivity patterns. You can see lots of networks are connected strongly to each other, with the exception of the somatomotor and ventral attention networks, who are a bit lonely. And we noticed that some of the networks that were extracted in MEG bore some similarities to the ones that we found in fMRI. They were certainly different as well, but there were some notable similarities. So the key question here we're interested in is, does an individual's expression of each one of these connectivity patterns, is it a function of the degree to which their symptoms of ADHD have persisted from childhood? And indeed, there was some evidence. And here I show that there were connectivity associations that emerged primarily between adult inattention and the expression of 4 of the MEG connectivity patterns and 1 of the fMRI connectivity patterns. And here you see one of the connectivity patterns that was associated with adult inattention. This one was in MEG, this is in the gamma band. Details don't matter, but it's a heat map that just shows how strongly connected the different networks of the brain are to each other, how strongly they're talking to one another. And here are the results for the remitted group, and you can see it's almost indistinguishable. And here are the results for the persistent ADHD group, and you can clearly see the dissimilarity, and this was a highly significant difference. The same theme was found across all 4 of the symptom-associated connectivity patterns that we detected in MEG. Throughout, the remitters did not differ from the never affecteds, but the persisters did. I'll note that no connectivity differences were found when we reanalyzed the data based on childhood history, comparing those who ever had ADHD against those who never did. So to recap, we find that remitters were essentially indistinguishable from the never affected in patterns of intrinsic functional connectivity as measured by MEG. Anomalies were confined to those with persistent ADHD. We did not find that a childhood history of ADHD left fixed anomalies, and there was no evidence of neural reorganization in the remitters. Note that this pattern of findings held when we removed those adults who were on current regular psychostimulants, and also when we excluded the 10 to 30% of those with current comorbid disorders. So, just to recap, by far we find that remission is associated with typical white matter microstructure and typical patterns of cortical intrinsic connectivity. However, this study focused on connectivity as defined by MEG. MEG isn't great for probing deep brain regions, subcortical brain activity, and this is important because, as I mentioned earlier, a very influential model by Halperin and Newcorn argues that subcortical anomalies is sort of where it all starts, and anomalies here will persist into adulthood, and we may well have missed them with this study. So in the final study I'm going to tell you about, we used fMRI as it's very good at probing subcortical activity, particularly during cognitive tasks. And the cognitive task we used was response inhibition, as this is one of the most consistently implicated cognitive mechanisms in ADHD. The paradigm that we used was the stop signal task, in which a subject responds to a stimulus unless a warning signal suddenly appears, and then they have to stop themselves. The task is adaptive, and it keeps the subject just on their inhibitory edge. What's nice about this task is it reliably activates both the prefrontal and the caudate, the striatal, deep regions of interest. There was around 180 participants in this task. They completed either fMRI, MEG, or both. So first, we conducted hypothesis-driven analyses on the fMRI data based on Halperin and Newcomb's model of remission. So first, we're asking, does cortical activation, specifically of the inferior frontal gyri, does it reflect adult outcomes? Secondly, will caudate activation reflect a childhood history of ADHD, showing fixed anomalies regardless of adult outcomes? And I'll just mention the data were pre-processed and cleaned as before. So first, at a behavioral level, we found that the remitted and never affected were similar, whereas the persistent made slightly more errors in the task and had slightly less efficient inhibitory processing, reflected in a slightly longer stop signal response time. So next we looked at the fMRI data, and in our region of interest analyses, we found that activity in the cortical region, the inferior frontal gyri, did indeed reflect adult outcome. Now this is shown here for the contrast between failed and successful inhibition. So if you just look at the graphs, the overall group difference here is driven by the persistent group who were hypoactivating, as you can see in green. The other two groups, the remitters in orange and the red, did not differ significantly from one another. No differences emerged at the cortical level when we reanalyzed data based on childhood history. And next we looked at the subcortical region of interest, the caudate, and here the inverse pattern held. So there were no significant differences between groups defined on the basis of adult outcome. Instead, the differences emerged when we analyzed on the basis of childhood history. Those who had childhood ADHD showed reduced right caudate activity when successfully inhibiting. So in short, using this strong cognitive probe of caudate function, we found for the first time anomalies that reflected a childhood history of ADHD rather than current adult status. Finally, I'll note a very interesting side finding is that in whole brain analyses, completely unconstrained by regions of interest, the most strong finding was actually cerebellar activity during a successful inhibition reflected adult outcome in the regions that you can see here. This was, to me, an unexpected, but I think entirely fascinating finding that actually we also find in MEG. And to talk about MEG, we followed up our fMRI findings using MEG, and we did so because it can parse cortical inhibitory processes with really exquisite temporal resolution. During successful inhibition, we detected associations between adult symptoms of hyperactivity, impulsivity, and neural responses in one region of the brain, and the region of the brain lay near the right inferior frontal cortex, which is shown just there. Activity in this region is shown in the graph below, and this shows gamma activity. So look first at the blue line, which shows, actually this is theta band activity in the remitters. So that's what the theta band is doing, the neuronal activity in theta in that brain region in the remitters in blue. The typical group looked very, very similar. Now look at the persisters. You can see there was generally less activity, and this difference reached significance during one narrow time window, which is shown in gray. And that time window is 300 to 350 milliseconds after the stop signal appears, which is just whenever successful inhibition should occur. So in short, using MEG, we found that during an inhibitory time window, individuals with persistent ADHD showed decreased activity compared to remitters within the right inferior frontal cortex. So in summary, for cortical inhibitory processes parsed using both fMRI and MEG, remitters did not differ from those who were never affected. By contrast, caudate anomalies using fMRI reflected childhood history, not adult outcomes, being present in both remitters and persisters. I'll note that these findings survived the removal of those who were currently taking psychostimulants and the exclusion of those with current mood and anxiety disorders. So how can I summarize this work? Well, overall, we didn't find much evidence of neural reorganization. For example, we didn't find that the remitters recruited brain regions during response inhibition that weren't seen in the never affected. We did find that the remitters frequently resembled the never affected group, compatible with the convergence model. This occurred at the cortical level, both in intrinsic functional activity and during inhibitory processes. And remitters also showed very similar white matter microstructural properties to the never affected. However, there was also some evidence of fixed anomalies that reflected childhood history at the caudate level. Now, in all of this work, we've benefited greatly, my group, from the conceptual and empirical work of other groups. Many of the teams are here, and here I highlight just 5 of the studies. All of these had exquisite prospective clinical follow-up and imaging conducted mainly at the end point of the study. I don't really have time to do them all justice, but I'll try to summarize them within this framework. So several of these studies find that remitters and never affected show similar prefrontal cortical function. This held true for 2 fMRI studies from different groups of prefrontal activity during cognitive challenge, and 1 EEG ERP study examining cortical indices of response preparation and vigilance. Additionally, remitters and the never affected showed similar intrinsic connectivity within the default mode network in a study from MGH. Is there any evidence for fixed anomalies found in both the persisted and remitted groups? Well, indeed there is. Perhaps most pertinently, Clerkin from Halpern and Newcomb's group found a typical hypoactivation in thalamic striatal regions during response preparation in both remitters and persisters. And interestingly, Cortese from the NYU group found some focal changes in white matter microstructure that were shared by persisters and remitters. Currently, there's probably the least support for the compensation model, but again, we're still at a very early stage. So, just to conclude, I'll again stress we are very, very far from a final answer to all of this, but at a cortical level at least, remitters do look somewhat similar to those who have never been affected. And remission seems to be able to occur even in the face of enduring subcortical anomalies. And it's important to remember that these different models are not incompatible. Just as we have multiple cognitive pathways to the onset of ADHD, there may be multiple neural pathways to remission. And indeed, remission within one individual might be tied to different, distinct developmental processes in different brain levels. And to echo what Adriana said, further advances here really rests upon acquiring better data. Specifically, the biggest challenge here will be the prospective collection of clinical and neuroimaging data that really embraces the comorbidity of the disorder. And on that note, I'm just going to thank my team. I'd like to particularly highlight two of the postdocs who did these wonderful analyses, Gustavo Sudra and Esther Sike, and also colleagues from NIMH, particularly Wendy Sharp, who's a clinical social worker, who's been with this study since its inception, and has been absolutely great, and Ellen Lieberluft, and the imaging teams at NIMH. And I'd like to thank you for your attention.
So, impressive studies. Going back over this whole debate about cortical versus subcortical, as Halprin first argued it, does the data support that or not? Or can you think of it in that kind of way? Okay. Where are we now in relation to that original hypothesis?
So, this very lovely model by Halprin and Jeffrey Newcomb, can you hear me? It posits that the onset of ADHD is a caudate event, and it will so caudate anomalies are quite likely to persist even into adulthood, whereas the clinical course may be more to do with cerebral plasticity of the cortex, and so that will more likely reflect adult outcomes. The adult endpoint studies, as again I stressed, because you're looking at the adult endpoint, you have to infer what's carried forward from childhood. So I think they're compatible, very compatible with this model, and it's certainly what we find. The clincher will be the prospective data as we map in tandem clinical outcome along with the imaging data that will really be needed to clinch this. And once we have the longitudinal yoked clinical and imaging data, that will provide us the opportunity to really test these models of remission. And perhaps more importantly, it might allow us to start parsing using methods such as cross-lag analyses, sort of quasi-causative mechanisms to really highlight the mechanisms that might be most important.
May I just add something about the subcortical-cortical? It may also be, and not to confuse to mud the water, it may also be that that idea does not just apply to ADHD. We have been seeing consistent subcortical-cortical atypical connectivity in ABIDE, in autism versus controls. The abnormal degree centrality in the lentiform nucleus coded was the transdiagnostic between autism and ADHD. So maybe that model could be become like a window for neurodevelopmental condition more broadly, and then it could be, when we think about the different pathways. I looked at the ABIDE data, and thalamus and striatum cortical connectivity is all screaming to say, hey, look at me more carefully. So I think it was a very intriguing model and very intriguing.
The cerebellum, but yeah, over here.
Thank you for these very informative talks. For Dr. Shaw, I have a question if you have any information about the childhood history of consistent, inconsistent, or none at all medication use, and what that might be doing either directly to brain function or in terms of allowing the space for learning while on medication to then impact brain function for remitters and non-remitters.
Yeah, no, that's a wonderful question about what role does medication histories have on all of these brain findings. So in the long term, I can tell you what we did, and then I can tell you what we ideally would do. But what we did was, for the longitudinal anatomic study, we calculated the proportion of time in the study that the child was regularly taking a psychostimulant. We had reasonably good records and could do that. For the adult endpoint study, that was that was a little bit trickier for some of them. So there we just decided to go with excluding those who were regularly taking medication and not. So that's one way. It's like treating a psychostimulant medication as if it's a nuisance. Either you covary it out or you drop groups who are regularly taking stimulants. What the long-term impact of psychostimulants are, absolutely fascinating, particularly on the brain. Of course, the ideal design is the impossible one. We'd have to do a randomized trial over a very long period of time, comparing those on medication and those not. In lieu of that, we have to do observational studies, just being as mindful as we can of not interpreting and over-interpreting any association, because it could always be due to a third factor. Those who have more severe symptoms may well be taking more medication, may well have poorer outcomes. But it's a great, great question, and an important one.
Yes, interestingly, my question was about the same. I remember going to a lecture, and I wish I could remember her name, but I can't, but she was a PhD at Michigan State, I remember, and she had done a study, I think it was at a CHAD meeting. She had done a study where she had measured data flow or neural messaging through the anterior cingulate. And she converted her data to a bar graph. And here was ADHD, here was, here was normal folk, here was ADHD, and halfway in between was people who had been previously treated with stimulants. And there was a strong implication that neurogenesis had been taking just the use it or lose it theory about neural function, and the more you stimulate neural pathways, the more they have synaptogenesis and increase. And there was some corollary data that actually increased levels of tissue. So I just remember that, and I have always wondered why, if that has some truth to it, why it's not more carefully studied, why it isn't talked about a whole lot, why the drug companies don't go crazy with it and try to promote it as having its positive developmental impact on a developing brain. So, and I know the anterior cingulate didn't get talked about much, frontal lobes didn't get talked about much when you were talking about your data, so those are my questions.
Comment on?
Yeah, no, do you want to? No, again, it's a very important question. What I will say is that we have a fair knowledge of, in functional imaging studies, of the acute effects of psychostimulants. So a lot of groups have studied that. So during the acute phase of a psychostimulant being on board, perhaps not surprisingly, as a psychostimulant improves many aspects, but not all, but many aspects of cognitive function, the acute effects on brain function is to shift it towards a more normative pattern. Whether that holds in the long term, not sure. In studies that have looked at more cruder measures, such as brain structure, typically the biggest finding, I think the take-home from many, many independent cross-sectional studies, and one or two longitudinal, is that there is a difference. The bigger difference is between having ADHD and not. The contribution to that of stimulants is absolutely tiny. If anything, it probably nudges it towards a more typical range, but it's very, I would be very cautious in over-interpreting that, because it's observational. It could all be a third factor operating. But it's certainly something that's there are one very good randomized controlled trial that did neuroimaging within the context of a trial of psychostimulants. It's a Dutch group. It's a very nicely well-done study, and it's something that we should all be trying to do, but it is exceptionally difficult, particularly in the longer term.
Is the anterior cingulate an important player in looking at your imaging study?
Pardon?
Yeah, I say, is the anterior cingulate another prime area to look at?
Oh, yeah, very important. It was actually one of the regions that, yeah, it really is. It's one of the little regions. It was one
that
of
was
the regions.
legit.
David.
I would like to underline the importance of waiting somewhat for the prospective data. Well, we can't wait for it, but keeping our brakes on, because when we re-examine the Halpern and Newcorn hypothesis prospectively, which had the limitation of, like you, only measuring the cognition at the end and not the beginning, we found the same results. We found that our remitters were those with the strongest working memory function, but when we went back, they also had stronger working memory at the beginning. So there wasn't change, it was actually predictive of a good response later on. And then when you add that to the fact that we and others, Phil's group and others, have found this dissociation between cognition and symptoms when we look at change, we mustn't make too many premature conclusions. So my question would be, how good are we now at thinking about the heterogeneity within brain structure and connectivity at baseline? Because it's not something that's been looked at, partly because of the small sample size. But if we started to do that and we were able to pass out heterogeneity early on, we could then start to think about whether this is predictive already or whether it's going to be change that's important.
Yeah, that's a brilliant question, because, and I like the reasoning of accelerating the pace of insights. I think that would tap in the approaches of that some groups are taking, including ours, of subtyping at baseline. So with the data we have, so data-driven subtyping, or at least your question makes me think about that. And I am not aware of multi-diagnosis subtyping yet. I'm aware of a few structural and functional, structurally based subtype confirmed and validated by functional connectivity patterns in autism because of ABIDE, because there were more than 2,000 data available that allowed to do that. Suk-Jung Hong has published with Boris Bernard. I'm aware of actually I am aware of a multi-diagnostic approach where they looked at latent factors combining ABIDE and ADHD 200, and they found in the default network they found common subtypes. But it's so initial, but I think you're right, that would accelerate already, and it goes back to my preferred model of unknown subtypes that maybe that do not really meet and read the DSM, but may have different trajectories and different outcomes. Yeah.
Thanks. Yeah.
So there have been these recent GWAS studies that have identified number of loci and potential candidate genes. Is it possible to go to your sample, and genetically analyze and see if any of those SNPs are related to the changes you've seen, or is the effect of the SNP so tiny that with your sample it wouldn't even be fruitful to do?
Yeah, what a nice question. So the GWAS is a great question, and actually we did that. That's exactly, and we did it first of all. So the GWAS, the big psychiatric genome, is a GWAS of the diagnosis. So we just looked at our kids, really, those who have ADHD, and we did exactly what you're talking about. We said, here is this individual's polygenic risk, common variant risk for ADHD. Here are their symptoms. How do they get there? So we used mediation and moderating analyses, and we looked at some of the, and actually, interestingly, Joel Nigg's group did this as well, and they found that working memory, a component of working memory, was one of the mediators, and we found that also. We also found some little things, and it's stuff that's in press, and it'll be out soon, hopefully. Great question about outcome. We know from some beautiful longitudinal twin studies in London that the genetic contributors to the outcome of ADHD overlap with those for the onset, but only by 50%. So it's going to be somewhat different genetic contributors. Given that half of the genes are shared with those for onset, you would expect polygenic risk to have a little bit of an impact on outcome, and we find a hint of that. It's not very strong, but then polygenic risk isn't. You know, it explains a couple of percentage of the variance. So we are finding that it's not even strong enough for us to then parse further into mechanism, but a good idea.
Yeah.
Hi. My question's for Dr. DiMartino. So given that we are seeing pretty different patterns of genetic etiology in ADHD versus autism, so in ADHD we're really thinking about the polygenic risk, and there's a lot of room for G by E interactions accounting for some of that variance, versus in autism we're seeing 30% maybe more of those cases can be accounted for single gene disruptions, and more if you count CNVs. How, if you were able to do whole exome sequencing on these kids in your data, which I know would be difficult given the size of the data sets, how do you think that would affect your outcomes if you were able to account specifically for those single gene disruptions in autism, if you excluded them or looked at them separately?
Oh, that's a very good question, an interesting question that I did not think about. It's a design I did not have the chance to think yet about. I am tracking in the current data analysis, well, first of all, I don't do genetic study, genomic studies, but so I am tracking who else is in the family and what are the diagnoses in the family, but that's not sufficient. And from what I am understanding of autism is, yes, there's 20 to 30% of autism is explained by the summation of CNVs, but it's likely that there's a combination of common and rare variants. So we are not there yet. So to me, that knowledge gap doesn't help me in the design of the imaging, and especially because I'm not, you probably have thought about this more because you have formulated the question with some ideas in mind. I don't know if you have a suggestion for me. Maybe we should meet and think about the next set of study. Yeah.
We've only got 2 more minutes. A quick question from Tom and Jeff, and quick answers, because we do need to end in a few minutes.
A quick question about the sampling for the studies on autism. How much differentiation is there between those with IQ well below average and those with average to well above average IQ in the samples you're looking at?
Yeah, unfortunately, most of the studies, most imaging studies are, at least those in school-age children and above, are on IQ in the intelligence range. So it's hard.
I'm sorry, I
to
didn't hear. In what range?
We don't have low IQ. We don't study low IQ due to the request and the demands of following instructions in the scanner. We do have, at least for functional imaging studies, we do have samples. We are collecting in the lab sample. I did not talk about this during natural sleep, and that allows us to bypass the problems of cooperation and intellectual abilities and skills. But I don't have results yet on that.
And
For
Jeff.
functional imaging data.
Thanks so much. First of all, I wanted to recognize the outstanding work of Kurt Schultz from our group, and note that Kurt is the co-author of that paper with Jeff Halpern, and he presented here, of course, some work. And I wanted to ask you, Philip, whether I was so intrigued and pleased to see you present clinical vignettes right before you presented this elegant imaging study. And I wanted to ask you, because what you then told us about is what we found after the fact in those who recovered, whether you're starting to do some work and have any glimpses of information you can tell us at baseline. What can we say about what distinguishes course? Can we begin to think about that too?
Yeah, so the questions, what are the baseline predictors of how these kids did?
Are there any?
I can tell you, we looked at, one of the reasons I became so interested into remission and persistence is I looked through the literature and I just said, well, what predicts how these kids are going to do? Because I see these kids, one of the questions parents ask is, I'm just about coping. What's going to happen in 10 years? What I'm really worried about is after school. And I just said, well, I'm going to look through so I looked at the usual suspects, which were, gender, IQ, comorbidities, all explain very low amounts of the variance in outcome. And I thought, well, something's got to attribute to it. And then I looked at socioeconomic and social stressors. That's pretty minimal as well. I thought, well, what about genetics and the brain? You know, maybe there'll be something in there. Now, the brain imaging at baseline, good though it was, you know, this was done years ago using a anatomic scanner that we wouldn't use now, a 1.5T scanner. So we didn't find much that predicted outcome, and if it did, it predicted it with minimal amounts of percent of variance. So there wasn't really much in baseline that predicts it. My hunch is that if we capture the early trajectories of change in kids, that might be more predictive. So for example, if you chart how the brain is converging towards at a very early stage or diverging away from at a very early stage, will that be a much more powerful predictor of where this kid will end up in 8 years' time? And that's a study we're attempting. It's very difficult, mainly due to orthodontistry, because most of our kids end up with braces. But you know, we're still going to go ahead with it. But a great question at the minute, not many explanatory bases.
And thanks to all you guys, because it was a fantastic session.
Yeah, so I'd like to, thank you for a great symposium. Thank you.